Welcome to this session. Here we are going to discuss embryology. This is our introductory lecture. Embryology is the study of the developmental events that occur in the prenatal stage. This field complements other disciplines like biochemistry, genetics, medicine, physiology, and it is derived from the Greek word embryon, which means the unborn. Embryology has a number of branches. We have descriptive embryology, which deals with the morphological description of the different embryonic stages that happen during development. And here the descriptions can be for a specific species, for example the human species, or it can be different species. And in descriptive embryology you are looking at the different morphological changes that are happening. For example, at one point you have the zygote, the other point you have the blastomere. In the second branch of embryology, we have comparative embryology, which deals with a comparative study of the different animals or different species, embryology of the different species. So here you are looking at the different embryological stages, or you can look at a single embryological stage for the various animals. For example, you may want to compare the embryological stage maybe at week 16 for a rat, and then for a cat, for a human, for an ape, and then you look at the various changes that are happening at that particular stage. In experimental embryology, here the studies are usually to understand the fundamental mechanisms that are happening in development. We look at the various stages in embryology, for example fertilization, cleavage, gastrulation, the embryonic induction, determination, and then the differentiation that happens. And then in chemical embryology, this branch of embryology deals with the various biochemical, biophysical, and physiological techniques to understand the various embryological events that happen at molecular level. We are looking at the genetic regulation of the various embryological stages, and then we are also looking at the other growth factors that are involved in the embryology. In teratology, it is a branch of embryology, and this is concerned with the study of various malformations or birth defects that happen during embryology. And here we can look at substances or chemicals that actually cause these congenital anomalies. And these substances are known as teratogens. An example is thalidomide, which is linked to anomalies in internal organs and then also with the limbs. The history of embryology, it is a fascinating field that has been there for quite a long time. It is traced back to the times of Aristotle, around 300 BC, where there was a shift in the study of embryology. Other than just believing in superstitions, there was a shift to making observations in what is happening in the embryological development. The other scientist that we honor in this field of embryology is Galen, who played quite a role in advancing embryology. And the other scientist is de Graaf, around 1672 AD. He was able to describe the ovarian follicle. And then we have Ham and his colleague Leeuwenhoek, who also around 1677 were able to identify the sperm cells. Stages of development. We have fertilization as the first stage, where there is fusion of the male and female gamete to form the zygote. And then in the second stage up to the fourth stage, this actually considered the stages of embryogenesis. And in the second stage here we have cleavage, where we find there is rapid mitotic division without increase in cell size. And then we have gastrulation, where there is development of the three germ layers, that is the ectoderm, the mesoderm, and the endoderm. And then we go to organogenesis, where there is development of the various organs in the body. And then lastly, maturation. Fertilization, as we have said, it is a process of fusion or union of the spermatozoa and the mature ovum, and this results in formation of the zygote. After formation of the zygote, we have embryogenesis, where there is formation and development of the embryo. And we have said in embryogenesis there are three stages. We have cleavage, where there is rapid mitotic division without cell growth. And then gastrulation, where we are going to end up with the three germ layers, the ectoderm, the mesoderm, and the endoderm. And then after gastrulation we go to organogenesis, where there is the production and development of the organs. In this illustration we are seeing the various stages that happen in fertilization. We have the sperm undergoing the acrosome reaction, and here it will be able to penetrate the corona radiator. So after penetrating the corona radiator, it goes ahead and it penetrates the zona pellucida. Once the zona pellucida has been penetrated, it will move and attach to the plasma membrane of the oocyte, where it will fuse with it and be able to release the genetic material. And at this stage we can say fertilization has taken place. So after fertilization has taken place, we end up with a zygote. So the zygote will undergo mitotic division to form a blastomere, and this usually takes about 30 hours after fertilization. So this blastomere will continue dividing. It will divide into 4 cells, and then into 8 cells, and then into 16 cells. And the 16 cell stage is what we call the morula. And this morula resembles a mulberry. So the morula will spend about 3 days, which is 72 hours, in the uterine tube, and then it will enter into the uterine cavity. This is an illustration for formation of the morula. So the zygote is here with the two polar bodies. Will undergo mitotic division to form the blastomere, the two cells stage, and then it will further divide to give us the 4-cell stage, and then to 8 cells, and then finally to 16 cells without change in the cell size. And this stage is called the morula. So once the morula enters the uterine cavity, it is going to float freely in the uterine cavity for about 2 days, and here it is going to be covered with endometrial fluid and mucus. So this fluid is going to be absorbed by the zona pellucida through the canaliculi, and once it is absorbed, it is going to accumulate in this morula. This is going to form a cavity, and this cavity is going to pull cells to different sites, so where we shall end up with an inner cell mass of cells and then an outer cell mass of cells. And this new uh, structure that is formed is called the blastocyst. So once the blastocyst is formed, we are going to have the zona pellucida becoming stretched and thin until it finally disappears, and this is usually prior to implantation. And then the cells are going to be in two groups. Here we have the inner cell mass, and then we have the outer cell mass. So the inner cell mass is going to develop later into the embryo, and then the outer cell mass is known as the trophoblast. So this is the inner cell mass of cells which will form the embryo, and this is the outer cell mass which forms the trophoblast. So the blastocyst uh, gives, as we have talked about, it's going to give rise to the trophoblasts, which is the outer cell mass, and these trophoblasts will later develop into the placenta and the chorion. And then the inner cell mass is going to give us the fetus, the amnion, and the umbilical cord. So for the trophoblast, it is going to differentiate into two parts. It is going to give an inner layer, which is known as the cytotrophoblasts, and then the outer layer known as the syncytial trophoblasts. So the syncytial trophoblasts are going to proliferate, and it becomes a multi-layered, multinucleated protoplasmic mass. And the cytotrophoblasts will also differentiate into a layer of the primary mesoderm.